So maybe we'll get started. It is my great pleasure to have the chance to introduce men who need to know introduction, Kirk Lohmueller of our own department. Kirk got his Bachelor of Science in Biology at Georgetown, then a PhD at Cornell working with Andy Clark and Carlos Bustamante. He then went on to Berkeley, where he did a postdoc with Rasmus Nielsen, and he came to join us here in 2013. Along the way, Kirk has piled up many honors. I'll pick it up at the postdoc level. He first got an NIH postdoc and then sloughed that one off after a while to take a Miller Fellowship at Berkeley. Since then, since coming here, he's been a Searle Scholar, an Alfred P. Sloan Fellow, a Hellman Fellow. He's gotten funding from the National Institute of Justice, from NSF, from NIH. He's recently gotten an R35 award, a prestigious new award for promising up-and-coming investigators to fund their general research program, which is great. Despite Kirk's relative youth, he's got a very impressive track record, which he helped himself by getting started as a high school student. So going back, his very first few papers were from work he did as a high school student, including a first author paper in Nature Genetics, which is bad. And over the years, he's piled up, he's up to 50 papers by now, and he regularly is hitting all the top-notch fancy ones and all the serious disciplinary ones, so acquitting himself quite well in the publication department. Kirk has built a very vibrant group here in our department, and he's really established himself as a leader in developing statistical and computational tools to think about genetic and genomic data. And in particular, I think he's really risen to the top of the field in terms of understanding the role of natural selection in shaping genomic data, and particularly deleterious mutations. And I gather we'll be hearing more about that now.
All right, thank you, Jamie, for that really kind introduction, and I'll hopefully live up to that today. But it's great to be here talking to all of you. So when new mutations occur in an individual's genome, they may be subjected to natural selection. There are a number of different types of selection that could affect mutations. One type is what's called positive natural selection. This occurs when a mutation arises that increases the reproductive fitness of an individual that carries it. And if this type of mutation would become established in the population, it may increase in frequency such that everybody in the population would carry it. We as evolutionary biologists are very interested in these kinds of mutations because they underlie a lot of the adaptations that we're interested in studying, and so there's tremendous interest in studying this type of selection. Another type of selection is what's called negative selection or purifying selection. This occurs when deleterious mutations arise in genomes, and because individuals that carry these deleterious mutations have a lower reproductive fitness than individuals that don't, these mutations may decrease in frequency over time and be eliminated from the population. There's been tremendous interest really since the early days of population genetics at quantifying the relative contributions of these two different types of selection or of these evolutionary forces to determining which one is more important at impacting patterns of genetic variation across genomes. This concept has been formalized in something called the distribution of fitness effects, or DFE. So what is this distribution of fitness effects, or DFE? Well, it's essentially a histogram or distribution of the selection coefficients for new mutations that could occur in a genome. So here, this blue curve shows one hypothetical distribution of fitness effects where there are many strongly deleterious mutations, many neutral mutations, and fewer mutations that have intermediate strengths of selection. A second hypothetical distribution of fitness effects could look something like this, where now we have many neutral mutations as well as some weakly deleterious ones, but fewer strongly deleterious mutations. A third distribution of fitness effects could also include a proportion of weakly beneficial mutations. So I would argue that this is one of the most important distributions in, I would say, all of biology, but certainly population genetics, because if we understand what this distribution of fitness effects looks like, we can then have a good idea of how evolution might affect genomes and what happens to mutations when they arise in populations. So I would say this is really one of the holy grails of population genetics that we as a field are trying to understand. Now, there have been a number of approaches taken to study the distribution of fitness effects and quantify fitness effects of new mutations. One approach has been to use experimental evolution or mutation accumulation lines, where essentially organisms, typically bacteria, viruses, or yeast, or Drosophila, could be essentially kept in the laboratory, evolved for a number of generations, and then the fitness can be measured at the end of this. And what comes out of these studies is that typically, especially in stable environments, the organisms at the end of this process tend to have lower fitness than the original starting material. And so what that suggests is that many mutations that occur in genomes tend to be deleterious. In other words, deleterious mutations are a ubiquitous feature of populations and genomes. And so for this reason, as well as several others that I'll tell you about in a few minutes, my lab at UCLA over the last 5 years has really taken a strong focus on understanding patterns and processes related to evolution of deleterious mutations. So I'll tell you a bit about why I think it's important to study deleterious mutations. So first, many of the evolutionary questions that we're interested in either directly or indirectly focus on what's happening with deleterious mutations. So first, if we're just interested in understanding genetic variation or the evolution of genomes, if deleterious mutations are commonly occurring, well, then they need to be an important part of understanding the genomes that we see in populations. Second, if there's interest in studying the molecular clock, or this clock-like accumulation of substitutions between species that occurs over time, well, typically there are assumptions made about whether mutations are neutral or whether they're under selection, and so understanding whether or not the molecular clock holds will be dependent on understanding deleterious variation. Further, if we're interested in understanding other processes, such as recombination, well, it turns out that the conditions under which recombination may evolve depend on the input of deleterious mutations in the population. Now, maybe you don't care about deleterious variation, but you care about adaptive evolution. And either quantifying it or finding adaptive mutations is very heavily dependent on having a good null model, or good model for the background evolutionary process. And I will argue in my talk here today that that's dependent on having or including deleterious mutations. Lastly, a topic that's, I think, particularly important for this audience, if we're interested in conservation biology or looking at the accumulation of deleterious mutations in small populations and genetic load of them, that's again very intimately related to deleterious mutations. Lastly, many questions in medical genetics these days also pertain to understanding patterns and processes surrounding deleterious mutations. So for example, knowledge about deleterious mutations will help us inform and deduce the architecture. In other words, whether rare or common variants are more important at accounting for risk for complex traits, and that in turn can suggest optimal strategies for detecting variants associated with disease risk. So in my talk here today, I'll tell you about 4 different projects in my lab aimed at understanding deleterious mutations and what happens to them in populations. And so I'll begin by discussing our work on the distribution of fitness effects for new amino acid changing mutations and understanding some of the biological factors that underlie this distribution of fitness effects. So first, my group takes an approach different from that experimental evolution approach I outlined in the introduction. What we do is we focus on patterns of genetic variation in natural populations. These patterns of genetic variation, say from sequence data, are the outcome of the evolutionary process, and they were influenced by forces like mutation, recombination, demographic history, as well as natural selection. And then we build evolutionary models that include natural selection to try to determine what component of this variation is driven by selection. So I'm going to introduce one of the key summaries of the data that we use. This is what's called the site frequency spectrum, or SFS. And so imagine here we have some DNA sequence data from five chromosomes. The variable sites are shown by these different columns. And this site frequency spectrum is just a histogram here, essentially summarizing the frequencies of the SNPs in the data. So for example, there are 5 sites or 5 positions in the data where the mutant or derived allele, denoted in red here in the sequence data, is present at frequency 1 out of 5 in our sample. And so these 5 sites are denoted by the 5 blue boxes in the figure. Similarly, there are 2 sites where the derived or mutant allele is found at frequency 2 out of 5. 2 sites where the derived allele is found at frequency 3 out of 5, this one, and 0 sites where the derived allele is found at frequency 4 out of 5. So the idea is we can easily compute this site frequency spectrum for really any compartment of the genome and any sample of genetic variation data from a population. So why might this be a useful thing to do? Well, what I'm showing you here is a cartoon. This is from a population genetic model here of what the frequency spectrum looks like from a standard neutral population. And then as we move from left to right, mutations become more and more deleterious. And so what you can see is as we move from left to right, you notice two things. One, the heights of the bars are all just smaller. And that means that there's less genetic variation for when we think about deleterious mutations. Second, of the variants that are still present, they tend to be more at lower frequency. In other words, they're skewed to be more at low frequency, and all the high frequency ones are gone. This intuitively should make sense because these deleterious mutations have been eliminated from the population and from the sample here by negative selection. So the idea is what the frequency spectrum looks like under selection is determined by the selection coefficients of the mutations as well as the demographic history of the population. And so one of the challenges to using this approach is, right, other things can influence what this frequency spectrum looks like, namely the demographic history of the population. So the approach that we and others have taken to circumvent this problem is to take a class of putatively neutral sites. So these could be non-amino acid changing mutations or synonymous sites if we're thinking about coding regions, and it's fit a demographic model to those sites, and then conditional on the demographic model, infer the parameters of a distribution of fitness effects for the amino acid changing sites that are influenced by both demography and selection. In other words, what are the parameters of selection that will be required for a given demographic history that gives you this site frequency spectrum to really turn it into this particular site frequency spectrum over here. So we've developed a computational approach to perform this type of modeling and inference called FitDaddy. This was pioneered by Bernard Kim, an EEB graduate student in my group. And this program has several advantages, computational advantages over existing approaches in that we consider a variety of different distributions for the distribution of fitness effects. We have some tricks using lookup tables to allow more efficient optimization than previous approaches. And the key advance, or what this allows us to do compared to previous approaches, is to use data from larger and larger samples on the order of hundreds or thousands of individuals. And that will enable more accurate inferences of the distribution of fitness effects compared to previous studies. So just to show you an application of this approach to amino acid changing mutations in humans, in red here are estimates that we made using genetic variation data from humans on the order of a few hundred to a few thousand individuals. So the three different red bars here are for different data sets. The black and gray bars denote the previous state-of-the-art inferences of the distribution of fitness effects for amino acid changing mutations in humans. And what you can see here is that for the larger data sets we infer, so these would be really strongly deleterious mutations with a selection coefficient bigger than 1%, and these would be the essentially nearly neutral mutations where selection coefficients are either 0 or all the way up to 10 to the minus 5. So this is really neutral. This would be the strongly deleterious component. And so what we infer using the new approach and using larger samples of individuals, we infer there are fewer strongly deleterious mutations and more essentially nearly neutral mutations from the data. And so this is important because it means other downstream inferences that rely on the distribution of fitness effects can now be made more accurate, allowing for this larger class of neutral mutations. So that's what the distribution of fitness effects looks like when thinking just about humans. But what about other species? Does this distribution of fitness effects differ across different species? So to address this question, we took a comparative population genomics approach where we used polymorphism data from multiple species to estimate the distribution of fitness effects and test whether or not it differs across species. So this is the work of Christian Huber, a postdoc in my group. And we started by focusing on two very phylogenetically different species, humans and Drosophila, because there's actually a large amount of genetic variation data available from populations from these species. And I want to emphasize that for those of you who have some familiar with the population genetics of both these species, right, you know that they have very different demographic histories and very different effective population sizes. And we account for this in everything I'm about to tell you by fitting demographic models to the two species separately and allowing them to have very different demographic histories. So what I'm going to be telling you about is robust to these differences in demography between the species. And so given these demographic models, we essentially fit a distribution of fitness effects to the amino acid changing mutations in each of the two species. And so what we began by assuming that this distribution of fitness effects follows a gamma distribution, meaning the gamma probability distribution, and that has two parameters to it, an alpha shape parameter and a scale parameter. So what I'm showing you here in this first plot is essentially the likelihood surface for these two parameters, where our best estimate for the shape and scale parameters is denoted by by this blue circle, and it's given in the table down here. When we do this same type of inference using polymorphism data in Drosophila, we obtain these estimates for the distribution of fitness effects. And the combined log likelihood of the data from both of these is essentially the sum of the log likelihoods for the first two. And so, okay, so we have parameter estimates. They look kind of different. But what we then did was we said, okay, let's fit a restricted model where we constrain the shape and scale parameters to be the same between both of these species. And how well does that model fit the data? So that's what's shown in this last plot, and what's shown maybe more convincingly in the table down here, that the restricted model actually has a log likelihood that is significantly and substantially worse than the log likelihood where each of the two species is allowed to have its own distribution of fitness effects. So in other words, we can reject a model of the same distribution of fitness effects between species, and instead we find this DFE differs. How does it differ? Well, what I'm showing you here are our estimates of the proportions of new mutations for different fitness effects. Blue for humans, red for Drosophila. Gray is the combined or the restricted model. And what you immediately notice is that humans have a much higher proportion of strongly deleterious mutations, and flies have a much higher proportion of essentially either weakly deleterious or nearly neutral mutations. So the obvious question is, why is that, and what are the biological factors driving this difference in the distribution of fitness effects between different species? It turns out this is a question that's received a lot of theoretical attention over the years. A number of different models have been postulated. We evaluated those, and most of them actually did not meet various predictions or the predictions of those models did not meet various patterns we saw in the data, but one model did. And that's the model I'm going to tell you a bit about now. And that model is Fisher's geometric model. So what is Fisher's geometric model? Well, this is a conceptual model that essentially postulates there's a multi-dimensional fitness space, and there's some optimal fitness point here, and then a population will exist somewhere in this space depending on what its fitness is. So in this example, shown here, new mutations that increase the fitness of the population will bring it essentially closer to this optimal fitness. Those will be beneficial or adaptive mutations. Those other mutations that essentially decrease the fitness of the population would be deleterious mutations, and they'll move the population away from the fitness optimum. It turns out there's been a lot of theory developed dealing with this model, and essentially Fisher's geometric model is specified by a number of key parameters, two of which I'm going to highlight here. One is the complexity of the organism, which is essentially the number of independent phenotypes under selection, as well as the pleiotropy of given mutations. So pleiotropy here is referring to the number of traits that a given mutation affects. And so it turns out Fisher's geometric model makes a number of important predictions that we can actually test with our data. The first prediction is that more complex organisms might have, on average, more deleterious mutations than less complex organisms. The intuition for this prediction is as follows. Essentially, if you have a more complex organism, which in this model complexity is defined by the number of traits under selection, it's then more likely that a mutation will essentially disrupt or break one of these important traits in the more complex organism than in the simpler one. And so essentially we already found support for this first prediction by comparing humans and flies, but we wanted to expand this analysis to consider other species. So we looked at genetic variation data from natural populations of yeast as well as mice, and essentially reperformed our inference of the distribution of fitness effects in those species. And what I'm showing you in this plot here is our estimate for the average selection coefficient of deleterious mutations on the y-axis for each of these different species. Orange is if we assume a gamma distribution for selection coefficients. Green is a model where if we actually use a parametric form of the distribution of fitness effects that comes directly from Fisher's geometric model. The punchline is, regardless of which one we assume, the pattern is strikingly similar, where as species increase in complexity, we see on average mutations tend to become more deleterious. And so we interpret that to be supporting this first prediction of Fisher's geometric model. The second prediction of Fisher's geometric model is that less pleiotropic mutations might have a more skewed distribution of fitness effects. So what's the intuition behind that prediction? Well, the idea here is if a gene is less pleiotropic, then it may either have a very big impact on fitness if that trait that it affects is related to fitness, or it could have no effect on fitness if that one or small number of traits have nothing to do with fitness. And so you can see this large variance then in the distribution of fitness effects, and that's reflected by this bigger skew in the distribution. Whereas mutations in genes that are more pleiotropic, well, essentially then you're averaging over more phenotypes, more traits, traits, and so the variance will be smaller, and the distribution may look less skewed. So to test this prediction, we can just simply say, what does the distribution of fitness effects look like for different degrees of pleiotropy? Of course, estimating pleiotropy for different genes is not a trivial thing, and the approach that we took was to use gene expression breadth as a proxy for pleiotropy, with the logic here being that we can say, okay, some genes have a tissue-specific pattern of expression, meaning they're expressed in only one tissue, and the idea is those genes we take to be less pleiotropic, whereas genes that are more broadly expressed across many tissues would tend to be more pleiotropic. So the idea is we can categorize these genes into different categories and then re-infer the distribution of fitness effects for these different categories. And so here I'm showing you the results of that, and let me walk you through what this plot's showing, because there's kind of a lot here. Up top right is the data results for humans, bottom for flies. We also stratify by overall expression level. That's reflected by the 3 columns. And what I'm plotting then on the y-axis here is the shape parameter of this gamma distribution. The important thing you need to know here is that lower values of this shape parameter correspond to a more skewed gamma distribution of fitness effects. And so what you can immediately see, it's most clear down here in Drosophila, is that for genes that are tissue specific, across the board they tend to have lower values of this shape parameter, meaning the tissue specific genes have a more skewed distribution of fitness effects. In other words, the less pleiotropic genes do have a more skewed DFE, which is the prediction of Fisher's geometric model. The last prediction of Fisher's geometric model is that species that have a smaller population size ought to have a more weakly beneficial mutations. Why does that, what's the basis of that prediction? Well, if we have a small population size, as I'll talk about a bit later in my talk, that can lead to the fixation of slightly deleterious mutations, which in this model, fixation of slightly deleterious mutations will lead to the population moving further away from the optimal fitness because you have the presence of deleterious variation. Well, if the population moves further from the optimal fitness, that then creates the opportunity for new beneficial mutations to arise and pull the population back towards the optimal fitness. So fortunately, we can leverage or test this prediction by leveraging some of the previous theory developed from Fisher's geometric model published by Lourenco et al. And the key parameter here is essentially the very long-term effective population size. So this isn't what we typically think of as effective population size governing neutral genetic variation, but the long-term effective population size over which the distribution of fitness effects would have evolved in this model. And the estimates that we come up with here of this parameter for humans in Drosophila are, broadly speaking, within an order of magnitude or two, consistent with what we see if we look at polymorphism data from more contemporary species. And so we interpret that as essentially supporting this prediction of Fisher's geometric model. If you look a little more carefully at this, we make an interesting prediction here, and that is that we actually infer there's a higher proportion of new beneficial mutations in humans as compared to Drosophila. And that actually goes against some of the previous work on comparing amounts of adaptive evolution in these species that focus on different statistics or different ways of looking at the data. And this is something that we're absolutely following up on in ongoing work, and we'll see what comes out of that. So in conclusion for this part of my talk, the naive assumption that distributions of fitness effects are consistent across species, that doesn't hold. Selection coefficients on average are more deleterious in more complex organisms. We see gene expression relates very much to the distribution of fitness effects, where genes that have a tissue-specific pattern of expression show a bigger skew in the distribution of fitness effects than genes that are more broadly expressed. Our results suggest complexity and population size are important factors that determine the distribution of fitness effects in different species. And other models that I didn't have a chance to talk about, while they may apply to certain parts of the phylogenetic tree, like microorganisms, they're not really, and may not be, broadly speaking, not supported by the data, but they're not capturing these important features and important differences in the distribution of fitness effects across the tree of life. So now I'm going to tell you about a different facet of fitness effects of new mutations, and that relates to the dominance coefficients of deleterious mutations. So first, when I say dominance, what do I mean? Dominance refers to the fitness effect of a heterozygote for the deleterious mutation relative to that of the two homozygotes. So imagine here we have little a is the new deleterious mutation, and big A is the wild type, sort of ancestral most fit genotype. Well, if the fitness of the heterozygote is exactly midway between that of the two homozygotes, this would be an additive mutation, and the dominance coefficient H for this type of mutation would be equal to 0.5. If, on the other hand, the heterozygote has a fitness that's much closer to the wild type or the more fit homozygous genotype, then this deleterious mutation little a here would be recessive and the dominance coefficient would be less than 0.5. On the other hand, if the one copy of the little a deleterious allele is almost as bad as 2 copies, this would be a dominant mutation, where the dominance coefficient h would be much bigger than 0.5. So obviously, dominance is a really important parameter governing what happens to deleterious mutations in different populations, related to a lot of other key questions, such as genetic load, inbreeding depression, why selfing may evolve, and all sorts of other things, as well as various medical genetic questions. So what do we know about dominance effects of deleterious mutations? Well, it turns out the little that we do know comes from a small number of experimental evolution or mutation accumulation studies done in essentially Drosophila and yeast. And what these studies typically find is that mutations tend to be partially recessive. And sometimes these studies find a relationship between the dominance coefficient here on the y-axis and the selection coefficient, such that more deleterious mutations tend to be more recessive. So while it gives us some broad brush picture of dominance coefficients, these studies have a number of important limitations. So first is they're focused on strongly deleterious mutations that you can measure in laboratory environments. Do these estimates of dominance coefficients apply to more weakly deleterious or more nearly neutral mutations? I think it's entirely unclear. Second, they use sort of what I'm calling simple laboratory organisms, and certainly they're worthy of study and important to study, but do these estimates of dominance coefficients apply in other organisms? I've already told you that distributions of fitness effects vary across species, so maybe dominance coefficients do as well. Third, when you move organisms into the laboratory, right, that's not in their natural habitat, and so do these estimates in laboratory conditions apply in natural populations? And I think the answer is unclear. So given that, I don't think it's an overstatement to say that given how important dominance is, being one of the most central things in all of evolutionary genetics, we actually know very, very little about dominance coefficients for most deleterious mutations. And so that led to the work in my group. We're actually using genetic variation data from natural populations to attempt to estimate dominance coefficients for deleterious mutations. And like what I told you about previously, we're going to focus on using the site frequency spectrum as a summary of the genetic variation data, along with population genetic models, to test different models of different dominance relationships. The simplest model is where all mutations are additive, meaning here, like this orange line. A second model that we're going to look at is where mutations maybe have a constant dominance coefficient, but it's something other than 0.5. And the last model is essentially the most flexible one, where the dominance coefficient is somehow related to the selection coefficient. So that's this HS relationship, or the green curve here. And this is, again, work of Christian Huber and Aaron Durvasula, a graduate student in my group. And so you might say, well, okay, this sounds straightforward enough. Why hasn't anyone done this before? Well, it turns out estimating dominance coefficients from genetic variation data is actually quite challenging. And I'll attempt to illustrate the challenge here by this particular slide. So what this is, again, showing you is the site frequency spectrum for deleterious mutations with three very different models of dominance here. And if you look at this, what you'll see is, wow, those site frequency spectra look remarkably similar to each other. And the reason for that is this dominance parameter h is inherently confounded with the parameters of the distribution of fitness effects, meaning you can have different distributions of fitness effects and different dominance coefficients that give you the same pattern in the data that you see. So there's an identifiability problem with trying to disentangle these parameters from each other. And that's been a serious impediment to doing this type of work. The advance that we made was to say, okay, what if instead of considering a single outcrossing population, what if we also were to imagine we had data from a population that's largely selfing? Now, if we take these same models of dominance coefficients and fitness effects, but stick them essentially, so this is simulated data to be clear, but stick them in a population that's now selfing, you notice these frequency spectra now are very different from each other. And so that gives us hope that we can then disentangle the dominance coefficients from the selection coefficients and co-estimate both of them. And that's the key idea behind the approach that we took. So we applied this framework to looking at Arabidopsis as a model system for studying this, focusing on Arabidopsis thaliana as the putatively selfing species and Lyrata as the effectively outcrossing species. And again, we're focusing on amino acid changing mutations as the putatively deleterious category for this work. So what we did was we used a series of likelihood ratio tests to essentially see which of these models of dominance coefficients could best fit the empirical data. And so here what I'm showing you is that a constant H model where mutations tend to be recessive fits the data significantly better than the standard additive model. But more importantly, the model with an HS relationship, in other words, this type of green curve here, fits the data significantly better than either of these constant H models. So in other words, we find strong statistical support for a relationship between the dominance coefficients and the selection coefficients. What do the parameter estimates actually look like? Well, that's what's shown on this slide here. This black line shows our estimated HS relationship. And so the x-axis here is obviously selection coefficients. These are the more deleterious mutations. These are the more nearly neutral mutations. And what we see is across a large part of this parameter range, mutations really tend to be recessive all the way up until they become essentially nearly neutral. Only then do they start to become additive or even dominant. The blue bars here show the cumulative distribution function for the selection coefficients. So essentially, as you add in more and more of the mutations, you start to see this is the proportion of really strongly deleterious ones, and this is then the ones that are more neutral. Just to put some numbers on here, for example, we infer that about 15% of amino acid changing mutations in Arabidopsis are more deleterious than selection coefficients of 10 to the minus third and have dominance coefficients less than 0.025. So we find strong evidence that deleterious mutations tend to be recessive and evidence of this HS relationship. So you might ask, what's then the biological mechanism leading to this HS relationship and mutations being recessive? Well, it turns out that's really, again, one of these classic old school questions in population genetics that's really been controversial since the early days of the field, with some of the early giants in population genetics essentially arguing over this, among other things, where Fisher proposed an adaptive explanation, where essentially you have modifier genes that are under positive selection that then lead to dominance or recessive mutations occurring in other genes. Wright said, well, no, genes may be in metabolic pathways, and so you don't need selection for dominance, and you just get it essentially as an outcome of these metabolic pathways. We actually find our data is not consistent with either of these two models, and I don't have time to get into details, but happy to chat after. But the basic idea is then we said, okay, we need another model that can be more general and could potentially explain the evolution of dominance. And I'll tell you a bit about how we developed that model, but I need to show you some interesting pattern first. And that is that we essentially grouped genes by different functional categories or by different expression patterns and re-inferred this HS relationship, or the relationship between dominance and selection coefficients. And what we found is for catalytic genes, mutations in those tended to be much more recessive, whereas for structural genes, or just genes that in general have really high expression levels, mutations tend to be much more additive. And so what that told us is gene expression may be somehow directly tied to why we see mutations being additive or not. And so we then developed a new model where dominance essentially can arise as a consequence of different genes having different optimal expression levels. This model was extended from some earlier work by Hurst and Randerson, and here's essentially what the model looks like, where X is the amount of gene, or level of gene expression. F of X is simply the fitness at that gene expression. And so the fitness then depends on an intercept and scale parameter, as well as a cost to increasing gene expression. So the key idea of how this model works can be sort of shown in a couple diagrams here, where if you imagine we have a gene that has a low optimal expression level, the fitness function could look something like this blue curve here, where this would be the optimal expression level. A mutation that's homozygous, a deleterious homozygous mutation, would reduce fitness, or would reduce expression level, and then consequently reduce fitness down here. And a heterozygous mutation in this model we assume would decrease the expression level by 1/2. And because of the shape of the fitness function in this particular area, it really won't have much of a change in fitness, leading to essentially mutations being largely recessive. On the other hand, if we have a different gene that has a much higher optimal expression level, the fitness function could look something different. And now if we have a heterozygote mutation that decreases expression by 1/2, it may have a much bigger decrease in fitness, and mutations would be more additive. So that's the cartoon of how the model works. What we actually did was simulate with different intercept and scale parameters, simulate data under this model, and can find a few salient features by this model. The first is this model does indeed predict a relationship between selection coefficients and dominance coefficients. In other words, we see this HS relationship. Secondly, genes that have a higher optimal expression level, shown by these orange points, tend to be more additive or tend to be less recessive than mutations that have a low optimal expression level. And the predictions from this model agree nicely with what we see empirically in the data, where for structural genes shown in orange here, these are genes that have high expression levels. And we see that mutations tend to be slightly more additive than catalytic genes or even the genome-wide background distribution, which tends to have lower expression level. So in summary, this model postulates that you can see different degrees of dominance of mutations simply as a consequence of different genes having different optimal expression levels and selection maintaining the gene expression at those levels. And the predictions of this model are borne out by what we see in the data. So in summary, for this part of my talk, we've presented some of the first estimates of dominance coefficients from population genetic data. Most deleterious mutations we find are partially recessive, and those that are more strongly deleterious tend to be more recessive. And we've developed a new general model of dominance arising as a consequence of the costs and benefits of gene expression. So far I've told you a lot about fitness effects of new mutations. Now I'm going to tell you a bit about what happens to deleterious mutations when we actually put them in a population. And you might think that, well, deleterious mutations, they're bad, they'll get eliminated from the population by purifying selection, and you just won't see them. It turns out that that view, while largely true, is a bit simplistic, and deleterious mutations can actually persist in the population for some period of time. And whether or not that may occur is dependent on a number of different factors. The first is the strength of selection. So if a mutation is very strongly deleterious, it'll be quickly eliminated from the population. But on the other hand, if a mutation is only weakly deleterious, then what happens to it may depend on the demographic history of the population. In other words, there's a chance that these weakly deleterious or nearly neutral mutations could drift around in frequency in the population and actually persist for some time. And that's a phenomenon that my lab has been very interested in studying over the last few years. And today I'm going to tell you a bit about some of our work on looking at the behavior of deleterious mutations as a consequence of demography in the context of using a canid system focusing on domestic dogs. So we're going to use dogs as a model to study how population history can impact deleterious variation. They might say, why are dogs a good system for this? Well, it turns out dogs have a very interesting demographic history depicted by this cartoon here, where there was a domestication process. That process involved a reduction in population size or population bottleneck, as well as more recent breed formation, which again involved multiple population bottlenecks as well as strong inbreeding. And so the idea is we can look at genome sequence data from purebred dogs that have undergone multiple bottlenecks, as well as these essentially village or street dogs that have been domesticated but are sort of classic purebred dogs, and compare these genomes to what we see from gray wolves, and essentially look at how have these different demographic processes impacted deleterious variation. This is the work of Claire Marsden and Diego Ortega-Delvecchio from my lab. And what we did was we looked at whole genome sequence data from 90 canid genomes. We had a mix of high and low coverage genomes. For the low coverage ones, we developed and used statistical approaches that took into account the uncertainty in the genotype calling. And so I'll tell you a bit about some of the results from what we found from this work. So one of the things we did was we just looked at the amount of variation or heterozygosity in the individual genomes. So each point here denotes an individual genome. Red are the wolves, blue are the breed dogs. The x-axis here is neutral heterozygosity. So that's just a summary of how much genetic variation there is. And it's essentially more or less proportional to population size. And so what we find is, right, the wolves have the most genetic diversity, the breed dogs have the lowest, and the village dogs fall somewhere in the middle. This is entirely what we would expect to see and consistent with previous studies. And this reduction in diversity in the dogs is a consequence of the population bottlenecks associated with domestication and breed formation. Okay, the y-axis is what's more interesting here. And what I'm showing you is the ratio of amino acid changing heterozygosity to silent heterozygosity. And what this is telling us, so amino acid changing heterozygosity, these are at amino acid changing sites, they're putatively deleterious. Silent heterozygosity is putatively neutral. So what this is saying is given some level of neutral heterozygosity, how much deleterious variation do we see? And what you notice here is that this ratio is higher in the breed dogs and the village dogs compared to the wolves. So in other words, dogs have lower genetic diversity, but they have a disproportionately high amount of deleterious amino acid variation. And in fact, there's a strong negative correlation between these quantities. So the next question is, well, what can explain this pattern? Can population history combined with purifying selection and deleterious mutations explain this pattern. So to test this, we carried out a series of forward simulations, including different demographic models, different distributions of fitness effects. And just to put in a quick advertisement here, we developed our own forward simulation software here called PreferSim. It's available on our GitHub site, and we published a paper on it a year or so ago. It has some nice features that are good for these sorts of simulations. The punchline is that we indeed find that reasonable models of demographic history combined with models of distributions of fitness effects can largely recapitulate the patterns that we see in the data, suggesting that the interplay between purifying selection and demographic history is what's influencing this result. In other words, the bottlenecks associated with domestication and breed formation have impacted patterns of deleterious variation in the dog genome. Now that's focusing on a relative measure, right, for this ratio of non-synonymous to synonymous heterozygosity, and ratios are a little funny to interpret. So what about just the absolute number of deleterious variants per genome? And so we can simply look for, again, focusing on amino acid changing mutations, using a bioinformatic approach to predict which mutations are more or less deleterious. We see wolves have, on average, more heterozygous deleterious variants than do dogs. But for homozygous-derived mutations, we essentially see the opposite pattern, where dogs carry significantly more than do wolves. And then we focus on derived deleterious alleles. In other words, counting these twice, because there are 2 deleterious alleles per genotype, counting these once, we see breed dogs have significantly more derived deleterious alleles. Quantitatively, that's about a 2.6% more increase in the number of derived deleterious alleles per genome relative to wolves. We also did a more model-based assessment of the genetic load and differences in genetic load due to population history in these populations. I want to draw your attention to this figure here. We infer that essentially we simulate with the demography and selection coefficients and look at the load at the end. And we essentially find that there's an increase in load in the breed dogs relative to the wolves. This corresponds to quantitatively about a 3% increase, which is similar to the empirical estimate I just showed you. So in conclusion, dogs have a higher ratio of amino acid changing to silent heterozygosity compared to wolves. We estimate the additive genetic load is higher, meaning if all mutations are additive, load is about 1 to 3% higher in dogs compared to wolves. But if you believe the earlier part of my talk, where deleterious mutations may be recessive, then the increase in load is even higher. And I think there's some interesting features here where we see similar patterns in bottleneck human populations, where they have an increase in the number of homozygous derived deleterious genotypes and a slight increase in the number of derived deleterious alleles, suggesting even though the specifics differ, there's some common similarities about how demography impacts deleterious variation. They might say, oh, dogs, they're weird. You know, they've been selectively bred by humans. Does any of this apply to natural populations? And so to do that, or to assess that question, we used the California Channel Island fox species. This is the work of Jacqueline Robinson in a collaboration with Bob Wayne's lab here. And what we essentially did was look at whole genome sequence data from island foxes on these Channel Islands off the coast of California. And we found some interesting things. So what I'm showing you here is just a plot from a gray fox genome, just looking at how much genetic variation there is across the genome. And really, okay, there's nothing interesting here. That's sort of the baseline level of variation we see in a gray fox. Where the story gets interesting is if we play the same game, but now look at these different island fox genomes. So we have 2 genomes from one of the islands, the St. Nicholas Island. They're shown down here. These aren't bioinformatic artifacts, and this isn't plotting the data and using, invisible ink here. There actually is this genomic flatlining, or these large regions of chromosomes where we just see essentially no heterozygosity on these different island fox genomes. So there's a huge reduction in genetic diversity seen on these island fox genomes. And we thought this was unusual, but we wanted to say, how unusual is this? And so what Jacqueline did was essentially conduct a literature survey, pulling in data from multiple species where people had done sequencing. And that's what this histogram here shows, is just how much genetic diversity you see, with a couple different species highlighted. So including like the Yangtze River dolphin or African cheetah, they're shown over here. So these are charismatic species that typically we think of as having low diversity. They fall over here. And our St. Nicholas genomes have almost an order of magnitude lower genetic diversity than these other species that have very low diversity. And so this suggests actually these island foxes are so far one of the species with the lowest genetic diversity seen in any eukaryotic species. And so what this tells us is they've had very long, prolonged periods of very small effective population size. And so this is an ideal system to see what does that demography do to deleterious variation. So what we did was we focused on first amino acid changing or non-synonymous variants. And what I'm showing you here is that the island fox genomes have more homozygous genotypes for deleterious non-synonymous variants, as well as loss of function mutations. And if you, again, want to believe mutations are all additive and not recessive, we've done actually additional sequencing now of island foxes in red and gray foxes shown in blue here. And this is just looking at the number of derived deleterious alleles per genome. What you see is that there's a slight but very consistent effect across all the island fox genomes of there being a higher number of derived deleterious alleles per genome, suggesting that the small population size has led to an accumulation of derived deleterious variants. So in summary, for this part of my talk, St. Nicholas island foxes have the lowest diversity seen in any eukaryotic species to date. We see an increase in homozygous deleterious mutations on these foxes. We also see a higher number of derived amino acid changing deleterious alleles per individual. And an open question is, these populations have persisted for a long time. Why is that the case? Ongoing work, we think we may have some answers, but I'm not going to give a teaser now. So stay tuned for, I guess, subsequent seminars to hear more about what we think may be going on there. So in the last part of my talk, I want to focus on a slightly different question, and that is how, so far we focused a lot on deleterious mutations themselves. But it turns out deleterious mutations occur in the context of the rest of the genome, and they then can impact nearby neutral variation. And so I'll tell you about some of our work focusing on how that can happen. So first, by way of introduction, there's two ways that selected sites could impact nearby neutral variants. The first is what's called genetic hitchhiking. And so you can imagine here's just a stretch of the genome. These are different chromosomes. These are neutral mutations, pretty straightforward. But then a beneficial mutation arises in some genetic background. And if that mutation goes on to fixation and there's no recombination, it's going to drag with it this blue chromosome here. All the other surrounding genetic variation will get swept away. So you'll see this reduction in genetic variation surrounding this selected site. A second process is something called background selection. And it's a similar thing, except now instead of there being beneficial mutations, the selected sites are deleterious. And that's why they look red and sad and sick. And so if they get eliminated from the population, they'll also remove out the linked neutral variation that is essentially surrounding them. So this background selection process will also lead to a reduction in genetic diversity surrounding the selected sites. So there's been a lot of work at trying to quantify these effects across genomes in many different species. And it's pretty well established that this process I outlined occurs and affects genetic diversity within a species. So here's probably the earliest paper on this topic from Begun and Quadro in Drosophila, where what they did was they found this positive correlation between genetic diversity and recombination rate. And they interpreted this coming from selection because regions of the genome with a low recombination rate are then more likely to be linked to other selected sites. And through the processes I just outlined on the previous slide, those selective processes will reduce genetic variation. In other words, lower values on the y-axis. Whereas out here, regions of the genome with high recombination rate, they're essentially uncoupled from selected sites. And so you don't have selection reducing diversity out here. This effect is seen in lots of different species, analyzing data in different ways. And there's a large number of papers over the years, including some that I've done, that document this effect. But what about looking at divergence between species? In other words, looking at sequences between species. Here the effect is less clear. There's some classic theory in population genetics saying that divergence should not be impacted by this linked selective process. And however, the theory essentially is focused on the divergence that accumulates. So if this is the present day, it's focusing on the divergence that accumulates after the two species have split from each other. And it essentially neglects the contribution to this divergence from polymorphism in this ancestral population, and essentially assumes that because this is essentially comprises only a small amount of divergence, we can sort of ignore it, and it shouldn't impact things. So whether or not the theoretical results still hold when considering this ancestral polymorphism is unclear. Further, empirical studies on this question also tend to come to conflicting conclusions, with some finding evidence of selection and others not. So what we wanted to do was test whether neutral divergence is affected by selection. And this is the work of Tanya Feng, a graduate student in my lab. And so what Tanya did was take genomes from different species, different mammalian species, filter out exons and conserve non-coding sites, leaving the material behind that we think is effectively neutrally evolving. And then we tested for various patterns that could attempt to quantify whether we see selection at linked sites. So here's one result where we look at the human divergence between closely related species like human chimp or human orangutan, and we find that that correlates with recombination rate. So this would be a sign of selection influencing link-neutral divergence, where regions of the genome with low recombination rate see this reduction in divergence, presumably due to linked selection. We also wanted then to look at more distantly related species, like say human-rodent divergence. And to do that, we used a different metric where we divided the genome into windows and focused on, of a given window, how many functional sites are there? In other words, coding sites or that sort of thing. And the idea is regions of the genome that have a lot of functional content are more likely to have more linked selection. And so then if these regions have more linked selection than these, do we see that that amount of linked selection on the x-axis impacts the overall divergence? And indeed, we find that it does, where regions of the genome that have more linked selection have a reduction in putatively neutral divergence. So that again suggests that linked selection can impact neutral divergence even between distantly related species. We then looked at a third pattern in the data, where this one's a little bit more complicated, so let me walk you through it a bit more carefully. So previous work has quantified across the genome how much linked selection do we think there is. That's in this study from McVicker et al., and they quantified it essentially by their statistic called the b-value. So regions of the genome with low b-values have more linked selection or more background selection. Regions of the genome that have higher b-values have less background selection. So this is the amount of background selection that we see that they estimated from primates. And what we wanted to see is, well, does human-rodent divergence at all correlate with background selection in primates? Now, why is that useful to do? Well, if it turns out that human-rodent divergence has nothing to do with linked selection, right, which is the null model here, then it ought to show no relationship with background selection in primates, because these things have nothing to do with each other. Whereas, if background selection can influence human-rodent divergence, and it's been roughly constant over long periods of time, then regions of the genome that show more background selection in primates would also be predicted to show more background selection when looking at human-rodent divergence. And then we would expect to see a positive correlation between these different quantities. Turns out when we actually look at the data, what we find is the following, that indeed we do see a very strong positive relationship between background selection estimated in primates and human-rodent divergence. So these would be regions that have lots of linked selection in primates. Also, we see that there's a big reduction in human-rodent divergence, and that's again consistent with the idea of those same parts of the genome being hit by linked selection even in the human-rodent ancestor. So what we wanted to do then is see, well, can models of population genetic models recapitulate these patterns? We started with a simple neutral model where there's no linked selection, just to make sure that our statistics are unbiased and behaving in the right way, because a lot of the sort of normal assumptions are being violated. And what I'm showing you here are for the human-chimp divergence as well as human-mouse divergence, the correlation coefficient between these different quantities. Red are the observed values based on what I showed on the previous slides. And the histograms denote the distribution we would expect to see under neutral models of just demographic history. And you see that the neutral models don't really recapitulate this at all. We then added in plausible levels of background selection using the estimates from McVicker et al. And we then see that for certain parameter combinations, we actually can very nicely recapitulate the observed correlations that are seen in the data, suggesting that background selection could be a sufficient explanation to explain our patterns that we see. Mutation rate variation across the genome alone by itself can't contribute to these patterns. I'm happy to afterwards. We find that linked selection can impact divergence between species, and that sort of goes against some of the sort of cornerstone theory of population genetics that says that divergence is not impacted by linkage to selected sites. We were able to recapitulate these patterns using coalescent simulations, and I think the biological implication and importance comes in these last two, basically saying that if we're looking at levels of divergence and how they vary across the genome, linked selection is one of the factors that has previously been neglected but should be thought about. And where that actually really could matter is if researchers often use divergence across the genome to try to estimate local variation in mutation rate. And our results argue that some of that variation in levels in divergence across the genome is not due to mutation rate variation, but actually due to variation in levels of linked selection. So in order to have accurate estimates of mutation rate, this really should be built into a null model of evolution across the genome. So in conclusion, I'd like to thank the folks who did a lot of this work with me and the collaborators at UCLA and elsewhere, as well as my funding sources. And lastly, I'd like to thank my lab. It's been a real pleasure and privilege to have worked with you over the last few years, and I look forward to seeing what we'll be working on and what we'll do over the coming years. So thank you. Yeah.
About the first part of the EEB, when you talk about changing environments, and I wonder how much does one expect the environment to affect the nuclear purification? Particularly, if you take some organism that has been in the stasis for millions of years versus one where it changes all the time in the environment, what would you expect that difference?
Yes, that's a great question about changing environments. I have a few answers for that. One is we want to actually directly look at that question actually in the dog and wolf system. And so that's work that we have planned. We haven't done it yet. But in terms of, for these results that I've presented, we've actually fit extensions of Fisher's geometric model that essentially allows for shifts in the fitness optimum, which would essentially be corresponding to these environmental shifts. And we at least found for the humans in Drosophila that those more complex formulations of Fisher's geometric model didn't fit the data better than the sort of vanilla Fisher's geometric model that we focused on. So I think what that means is not that the environment hasn't shifted, but just that that's not driving a lot of the key features for overall genome-wide patterns of distributions of fitness effects. It would be interesting to focus on maybe specific categories of genes that might relate more to the environment or environmental shifts to see whether that same pattern holds, and that would be a really good thing to do. Yeah?
So I was surprised to see that village dogs and breed dogs didn't really differ in the amount of poetry variation.
Yeah, I have a couple thoughts. So you're absolutely right that in the scatter plot that I showed, the village dogs looked kind of like or pretty much like the breed dogs for the ratio of amino acid changing to silent heterozygosity. And so sort of two comments on that. One is that it's perhaps possible that that increase in relative to wolves was not actually due to the recent breed formation bottlenecks, but rather was driven by a domestication bottleneck that was much more ancient and shared by both the breed dogs and the village dogs. That's our favorite interpretation. The other caution, though, that I would point out is the village dogs had lower coverage data, and so the estimates were generally quite noisy for those. So I wouldn't read too much into basically that result as much, because the data is not as good. But that's something that we could follow up with better sequencing. Yes?
When you see a genome that has very low mutation rate, can you predict its past history of what kind of mutation the three types involve?
Yeah, so if you see very little genetic variation, that will tell you something about the demographic history of the population. For some things, having fewer variable sites may limit the resolution on some of the inferences. But yeah, you can reconstruct the history pretty well. And the lack of variation, like with the island foxes, actually does tell us something about that history.
That all three mutations are low, or what sort of?
Well, the idea is there's just low, so the mutation rate may not have changed, but it's more that the demographic history allowed for many of the variable sites to essentially be lost from the population because the population size was so small. Van?
You showed for the more complex organisms that inhabit those mutations are higher. I've seen studies in model show for the number of lethal mutations that's higher in simple organisms. So the question is, do you see that in your data? And also, Alan, question, sort of complex versus simple, define the number of genes or genomes and things like that. It's often tricky too. So you have any thoughts about that kind of complex versus simple category?
Yes. So I'll take the second one first, and that is that. Yeah, so complex and simple, right? That's a whole. We could have a whole hour seminar just on that. So I guess a couple answers to that. One is in Fisher's geometric model, there is a more precise definition relating to the number of phenotypes under selection. In terms of how we thought about it in terms of just regulatory architecture or just the essentially genomic, not genome size, although that's also been used as a way to think about it. But there are studies, for example, that actually explicitly looked at humans and flies and looked at how many protein-protein interactions do they have, and they find that even though the number of coding genes isn't that different, there's far more protein-protein interactions in humans compared to flies. So I think that's sort of the where we were, the level with which we were taking it. I would say, like, if we want to argue within mammals is one more complex than the other. I don't really want to go there, I mean, I think we can argue yeast and humans are a difference in complexity there. Relating, to your first question about lethal, that's with measuring very strongly deleterious. They're harder to measure because they're just generally not segregating in the sample. And our approaches gain a lot of power from saying what do we expect to see in terms of levels of variation, and then if we see less than that, then that tells us, well, it's been selected out. So we have a good handle on what's been essentially moderate, maybe neutral, weakly deleterious, and moderately deleterious. But to distinguish between moderately deleterious and lethals is actually quite hard to do with the size of these data sets. But I think as we start to sequence thousands and even more individuals over the coming years, we could more explicitly look at these very recessive, very strongly deleterious mutations. Brad, did you have a?
So in terms of the last part of your talk,
Right. Right.
So, do you have any comments on whether those
So that's a great question. And I think that would be something that could actually be fun to look at in a more situational, like in other words, for specific population sizes and divergence times. I would say typically I think it is true that the more divergent the species are, then this contribution from ancestral polymorphism does become smaller. So that's sort of the previous intuition, and I think that is true. What I'm saying is that it doesn't really quite go away, or it goes away. You have to be much more diverged than what people thought. So I guess it's hard to give a general answer other than we should look at it. But I would think let me give something a little bit more helpful here. So what the main effect of this selection would be to reduce the ancestral population size. And so that could in principle, it would probably compress the times, but it may actually improve things like incomplete lineage sorting and other sorts of things. So it could actually it could be beneficial in some contexts. Yeah, I absolutely think that would be a fun thing to look at. We should talk out.
Yeah. Question?
You mentioned structural gene dominate over a particular mutation. Why was that?
So it was the genes encoding structural proteins. The mutations in those genes seem to be slightly more additive than genes in enzymes. And our explanation for why we think that was the case was because the structural proteins tend to have higher expression levels than the catalytic genes. And so our model would predict that the genes that have lower optimal expression levels would then be more likely to be recessive in other words, the catalytic genes.
Okay. Well, thank you very much, Kirk.